Good afternoon. Thank you, Josh, and the organizers for the summit this year. It's certainly my pleasure to speak on behalf of the committee and introduce also to you some vascular analysis enabling technology developed by NASA. So my focus has been since about 1998 on vascular patterning after making some observations at the microscope that different cytokines involved key to this kind of processes such as angiogenesis, microvascular remodeling. Each cytokine such as VEGF, TGF-beta, inhibitors such as angiostatin and the steroids, each actually resulted in a distinct fingerprint sort of vascular pattern response. So this suggested to me the idea of developing the software where we first map and quantify these patterns, which are essentially fractal patterns in nature, so that these patterns could be described and provide informative readouts of these very complex signaling processes. In addition, I have the privilege of being the chair of the Vascular Imaging Computational Analysis Biosensing Committee, ICAB. Sorry, I was just trying to advance it here. I think I need a bit of help with the remote, sorry. So what you can see in the first slide actually.
Should just be this one. Yeah.
Oh, thanks.
So that'll go back, yep.
Thanks, Josh. And what you see in the first slide actually is an image from a clinical image of the human retina illustrating an arterial tree, a branching arterial tree. And on the other side, the right-hand side, a microscopic image that we've analyzed with the VestGen software from the mouse retina showing the vascular network geometry. These two types of geometries, as many of you may know, are the two basic types of vascular geometries in our bodies. Oh, it's actually very light, okay. So to introduce our committee to you, some of the members are here and perhaps they can stand up or wave in response. We have from the NASA Johnson Space Center, Lisa Carnell, Jennifer Fogarty, and Anthony Jeevarajan, who work in both cellular bioscience in space as well as the Astronaut Human Research Program for safeguarding astronaut health. We also have Krishnan Radhakrishnan from the VA Veterans Administration in Connecticut, and he is a long-term collaborator on the development and the applications of our vascular analysis program. We are actively recruiting members, not only members if you have yourself some kind of vascular biosensing or computational analysis imaging technology you think would be enabling, or if you just would like to make recommendations of people you know who have expertise in this area. So the other purpose for my talk this afternoon is to introduce you to the VestGen or vascular analysis software developed by NASA since I joined NASA in 2001. And it certainly is my pleasure and privilege to announce that NASA is supporting the public release of the software later this year. So really what I want to communicate to you is if you have some of these vascular analysis, quantification or patterning problems you wish to pursue, this will be available in the near future for your research. So to develop this analysis, it actually has been informed by what I consider nature, mirror matrix. Jeff Ross talked about that earlier today. It's informed by nature, and we use physiological branching rules for these vascular trees and networks to determine first the mapping and then the quantification results. I'm happy to demonstrate the software for you if you're interested during the remainder of our summit while we're together. It just takes a couple of minutes. I have to say, I think this is an example of how NASA can really work in, obviously not the wonders of space exploration, but in working collaboratively, bringing together engineering, computer science, biomedical researchers to work together collaboratively on this kind of software solution. So you see here an example. I thought this one might be an interesting one to show first. Essentially, we have a snapshot approach because of the time limitations. But this was a collaboration with a Harvard physician scientist who works in the GI and in cancer applications, who does superb intravital imaging in the GI system. So you see here the arrangement of a healthy intestinal vascular network, and you can see how regular, almost like a chain link fence, the geometry of this vascular system is controlled in a healthy state. But when you look at the bottom to the bottom left, in an inflammatory model, progressive inflammation, the vascular system breaks down and becomes very irregular. And then in the final column, what you can see is about a 50% rescue of the vascular morphology by using an over-the-counter probiotic. These results were correlated, as you can see, illustrated here with other histologic, immune, and cytokine research as the signaling mediated of VEGF by some of the receptors. So now, just to showcase a bit the vascular tree analysis, the point of this is not just to break down the system into different pretty colors, but the fact that the small vessels have a very different function from the larger vessels. So by mapping and breaking it down on a fractal approach that utilizes other branching rules determined by our cardiovascular physiology, we could make site-specific measurements of the small vessels compared to larger or medium-sized vessels. And this has really allowed us to make several surprising findings for both astronaut health in retinal responses to microgravity, but also in early stage progression of diabetic retinopathy, introducing a new window of regenerative opportunity that currently is supported by an NIH award with Dr. Maria Grant, who is also a member of our alliance and works in stem cell therapy. So I can only show, I suppose, this one slide about the specificity of the vascular patterning response. Here we applied a steroid that is a strong anti-inflammatory throughout the body, and you can see in the lower panel the response of the triamcinolone acetonide, the vessels to the steroid, introducing very pronounced morphology changes that we were able to map and quantify. But the patterning change is, as I said, very different from that would be induced by VEGF, FGF, or other therapeutics. And here what we did that may be of some interest for stimulating your imaginations for your own research, we looked at normal healthy heart development in avian and rodent models, first characterized the development of the coronary vessels from an amorphous network to a transitional network tree composite structure, and ultimately the resolution of these vessels into a mature tapering tree. And what we have been tasked by the Centennial Challenge, for which I very much consider Lynn Harper, our integration lead, a collaborator. She has asked us to compare the branching rules that we have found for the human retina with the branching rules for the superficial trees that you see on the surface of the brain or the dorsal side of the heart. Finally, what you can see here is some of the new revolutionary imaging that has been reported in recent years in the ophthalmology research conferences. What you can see now from these images is that for the first time in human history, we can now see every vessel in the human retina. And it is those small, previously invisible vessels that are so key in the progress of these blinding diseases, such as diabetic retinopathy and AMD. So here we were able to work with Dr. Richard Rosen from Manhattan, a vitreoretinal surgeon, comparing a healthy subject to the left, the Vestgen analysis of the tree, compared to a central retinal vein occlusion, a very obstructive kind of event for that middle-aged patient, compared to his fellow eye, which actually we could measure was in a state intermediate between the normal, healthy, younger subject and that diseased eye. So this is just a snapshot showing we've had a number of different tissue applications, experimental studies with different cytokines and regulators, as well as disease progression. And you can just hint at the complexity of the quantification that supports these kinds of mappings, as reported in our 19 publications, including an invited review paper. We've done some preliminary studies even in plant leaves or the fruit fly wing. But just to say that, again, in those applications, the branching rules of these fractal vascular structures present in all higher terrestrial life forms have really quite similar kinds of branching rules that nonetheless are, of course, organism specific. So I'd like to thank you for the opportunity to introduce and invite you to our enabling committee. I've summarized for you some of the VestGen applications, and again, my pleasure to say that this software will be released to the public in the near future, and very much to the Alliance members. Would like to thank and acknowledge my collaborators supported by the Centennial and other NASA programs, the scientists, the COTE scientists, David, Hamed, Rodney, and Nikunj, from Glenn, Mary Vickerman, Mark Lagatus from here, and then on the biomedical side, Matt Murray and Sophie Schreers and Candace Tahimic, and our undergraduate interns, Sneha, Marina, and Cassandra. So thank you for your attention, and if you have any questions, I'll be happy to entertain them now or later. Yes?
Does the tool allow differentiate between vascular genesis and arterial genesis?
So are you saying in the sense of new vessels that are not yet arterially or venous specified?
Formation of new blood vessels versus branching out from the existing arteries. Does the tool allow to differentiate between the two?
I
So
think,
they're two distinct mechanisms.
yes. Well, we would certainly be able to map and quantify those structures you're referring to, and then I think it is for the investigator to compare those results and describe what the differences are. But that raises an interesting point that scientists do ask, how predictive is this kind of an approach? And I think that's something we would do in the future. Right now, we're doing the mapping and quantification. We're becoming educated about this, and then that could be applied for more predictive applications in the future.